Everything is a chat, the same way that you talk with ChatGPT. Small molecule is docked on this protein. It creates a new protein. This is our Copilot. So for those of you who are not following all the AI trends, 2023 was the year of ChatGPT and Copilot. So with ChatGPT now you can do your own research, you can write articles, or you can write small novel, right? Or with Copilot, you can write snippet, you can write bigger blocks of codes. And these are becoming very popular and very standard. So we reimagine the molecule design world, and especially protein design. We thought we can do the same thing. You can basically design proteins and small molecules with this tool. So we reimagined how protein design will look like in this new world that we're going to. So we built this. We built a 310 Copilot. It's a very simple interface. It's web-based. You don't need to download anything. No plugins, no C++. You don't need to be a software engineer. It's just, I say it's as easy as use as Instagram. It's a web-based interface, and we brought all the tools into one page. Everything search, visualization, our own generative AI that I'll get into in a second. All the industry standard models, third-party models, open source models, they're all in one place. All the data science tools, all the reporting, etc. Just to get to the exciting part, I start demoing some random features in the product for the first demo. This is some of the features that we showed last week at NVIDIA. It's going to be more today. So I'll start with just everything is a chat, the same way that you talk with ChatGPT. You say, find the protein that binds to zinc. I'm an AI engineer. I'm not a bioengineer, so I just search something very random. Find the protein that binds to zinc and has size between 100 to 150. I pick a small 100 to 150 is the number of residues, number of amino acids. So I pick something small so we can, so everything is faster, and they're also more nice looking proteins to me. So these are the lists. I pick one of them randomly and I fold it. Can you fold Q, this guy? So again, it's a chat. It finds the right folding model, which would be the fastest DSM fold, 3 seconds on an A10 GPU. And this is your result, right? Pretty quick. You don't need to download the software, nothing. This is your result. And now you say, now I do something a little bit more fun. I search for a small molecule that is vitamin D. Again, everything that I do in this demo is very random. So I just want to show the feature. This is your small molecule. And now I want to dock this small molecule on the protein that we just folded. Obviously, biologically, this doesn't mean anything. It doesn't make any sense, but you can see what's going on here, right? It finds the right docking model, which is a DiffDock, the model that came out of MIT in 2023. It's running with 10 num cycles by default, and here we go. Now your small molecule, your vitamin D is docked on this protein, and the score is actually pretty bad. It's minus 2.67, which is basically, that means this is not a good binding. Again, very random stuff. So now, so far we just connected to a bunch of industry models. Now this is the fun part. Now I ask our own AI to diversify P1, and I give it a temperature. The temperature is basically a metric of how liberal the model can act, right? It goes from 0 to 3. At 0 means it actually is very conservative. It almost repeats the initial, the original sequence. If you go to 3, it just gets to a very crazy protein. Something around 1.6, it changes some of it, but it keeps the majority of the structure. So I diversify, so it connects to our own end-to-end generative AI model, the model that we trained on 40 million sequences of size less than 500. And as you can see, it creates a new protein that is somewhat similar, but the red parts, the red residues are the parts that we changed. Now you can say, okay, this is just a random painting that you showed me, and there's a random sequence. What's the significance of this? And these are very random, by the way. I run into problems here. I say compare P1 and P2 just to do some sanity checks, just to do some guardrails, make sure this is not a hallucination. P1, I say compare P1 and P2, and it connects to Prot NLM, which is a Google model. And there you go. As you can see, the tags and the features are very similar. So that's just some sanity check that shows the molecule that we designed is actually has the same properties than the original models. So the idea here is to everything that you need, every piece that you needed to download softwares or run separate models in different environments, everything is in one place. You have your chat, very easy interface to talk to. You have your visualization, basically, screen here. You can see your sequences, and you have access to all the industry standard models, and you have access to our own generative AI models that gets better and better every day, basically. So now you might say, okay, I want to do something a little bit different. I want to redesign P1, but I don't want to redesign all of it. I want to redesign just the beginning of it. So redesign P1 from 1 to 30, only the first 30 residue. And yeah, that's a simple thing. Okay, we do that. And again, these are all, we show our features in a random fashion, and there is no goal for this demo. In the next demo, that's the demo that it gets to cooler stuff. So this is, again, you can see it's somewhat similar, not exactly the same. 5 seconds on an A10 GPU. It runs our generative AI model, and you see the results. Now I say compare P1 and P3. So when you do the comparison, you are going to see the result of ProtNLM, as well as, and again, a lot of similarities. Again, you see the TMS score of 0.85, which is not that significant. We actually don't know if you want to have a high TMS score here or low TMS score. High TMS score basically means if our TMS score is 1, it means we just copied the original. If it's low, it means we diverge from the original. In different contexts, it could be positive or negative, whether we want to go more diverse or more similar to the original. But this is just a demo that shows how you can play with different tools in one environment, no specific reason.